to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 29th episode of the Weekly Bioanalysis. My name is Dominic Warino and I'm the Senior Scientific Advisor for all things related to ligand binding assays, biomarkers, and flow cytometry. I've been with KCAS for a little over 7.5 years now, but I've been in the bioanalytical space for close to 26 years. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, Senior Scientific Advisor with a focus on LCMS technologies. I've been with KCS a little over a year, but again, I've also been in the bioanalytical space for around 26 years.
Today's podcast, as always, is brought to you by KCAS, Bioanalytical and Biomarker Services. KCS is a bioanalytical laboratory located in Kansas City, Kansas, serving the pharmaceutical and biotech industries for over 40 years now. John and I are the senior scientific advisors at KCS, and either or both of us are available to answer any of your questions regarding this podcast or any of KCS services. The goal of this weekly podcast is to give you a chance to connect with all of us, and we were hoping it's a chance for you to connect and get to know us, and even more importantly, for us to give you a chance to get to know KCS and the services we provided. As always, the podcast will be a review of the latest news and resources, and then a focus of a topic of our choosing before discussing any feedback we've had. We're constantly looking for feedback, and we'd be happy to discuss something that you want us to cover. I should note that you can find the podcasts, our weekly podcast. This is our 29th one. They can be found at kcsbio.com. And as a reminder, you can also follow us on Twitter at The Weekly Bio and at KCSBio. If it's the first time you're tuning in, I'm here in the Kansas City area. My colleague John is in upstate New York, and our producer is in Missouri somewhere running wild. Hi, Jeremy. I hope you're doing well. Anyhow, as I mentioned, the podcast will focus on a main topic. Then we'll give you a little bit of a teaser for next week and let you know what is coming up in the future. So again, we're very excited to be here. We're looking forward to a fun episode. To kick us off for podcast number 29, John's going to go over the podcast topic a little more. John?
Yep, as usual, we'll go through the news items, which will touch on COVID, then some non-COVID news. And our main topic this week is the latest and greatest on drug delivery, essential part of where the drug development is going and where pharmaceutical companies have a lot of focus. And it's a fascinating touch on the technologies that are being explored to help us all.
Yeah, John, it's a passion that we've talked about on previous podcasts, but for now, let's just jump right into the news and notes. John, why don't you kick us off with our first COVID item? It's from Twist Biosciences, and it's pretty germane to today's topic. They're looking at nanobodies inspired by llamas, which show promise in hamsters. John, what is all that? Llamas and hamsters. It sounds like
I always like the nature-inspired medical topics. I mean, this is, it always appeals to me where you've got, scientists applying to develop drugs, etc., but they're still learning from nature, and that's what pushes things forward. I mean, a really good example of this is Byetta or exenatide, which is the anti-diabetes drug, which is basically a 39 amino acid peptide, and is a synthetic version of Exendin-4, a hormone found in the saliva of the Gila monster. That's the prime example, is the approved drug that's made it to market. But these nanobodies are inspired by llamas and other camelids, so it's not a full antibody, but it's a single domain antibody. So where your conventional antibody's got the 2 heavy chains, 2 light chains, with variable heavy and light domains. This is just a single chain, and the hypothesis is by being smaller, it can more directly access hard to reach targets, and this company Twist Bioscience are in preclinical. They've tested 2 candidates. I think they're synthetic nanobodies. Anyway, they tested 2 candidates against hamsters, and this is one thing I hadn't realized, but one of the readouts for for hamsters in terms of COVID-19 infections, weight loss. And so in this study, the nanobody treated hamsters didn't show weight loss where the COVID infected ones did. So they're showing these nanobodies, according to the article, showing promise. And they tested 3 dose levels, all of which showed an effect. So I assume it'll be additional, preclinical, and then moving on to phase 1.
Yeah, John, one of the key advantages of using a llama, apparently, and I did not know llama immunology until I read this, is that they, instead of a traditional heavy and light chain kind of 4 parts, if you think of it, for an antibody, meaning 2 heavy and 2 light chains to form this like binary or bispecific or sort of two binding domains of an antibody. This is just a single chain, and as you mentioned, it becomes much smaller, which has huge advantages in terms of allowing for movement into areas where normal antibodies might not make it in. And so this is also something. What I do know is we use ovid or bird antibodies, chicken antibodies, as blocking reagents because of what I just said. They're smaller and they allow for better blocking. So it's in the ligand binding assay space. We use it to improve our signal to noise ratio, John. So pretty cool stuff, really fascinating. Should also mention here a little bit, University of Vanderbilt Medical Center is involved, and they have a very strong COVID program. So really kind of cool stuff from Twist.
And just one other comment on the nanobodies. It may also help with delivery because you may be able to look at inhalation as a potential route of administration, things like that. So it does touch on the drug delivery piece that we want to talk about later. But there's a lot of potential advantages here.
Yeah, smaller is better in this case, but
Sure.
really cool stuff. And anytime you can talk about a llama, it's all good, right? Anyhow, John, maybe on to the next one. One of our
animals are cool things. All nature is just fascinating to me. So the more we can talk about it, the more I'm involved.
You already hit the Gila monster, which again was like a deep cut, John. Very impressed with that one too. I won't go over snake venoms and how they've impacted immunology. So,
don't get me started on snake venoms. I'll tell you about all my work.
Well, poison ivy. I've always seen that you can cure cancer with poison ivy. You just got to figure out how to mix it the right way. No, I'm just kidding. So, John, there's been we've talked about some of the ups and downs in farmer. The perception of farmer has increased or at least gone up. And now AbbVie has launched a full frontal campaign with 3 new ads, right? In the psoriasis, Crohn's, and rheumatoid arthritis space to continue to maybe ride the wave of the vaccine, the COVID vaccine that has really heightened our perception of big pharma. So,
I think the message I took from this is basically they're releasing these ads, but it's really to emphasize balance your medical care that you're already ongoing with the pandemic. So if you've got these diseases, you really still need to be, getting to the doctor where you can, whether it be through virtual appointments or any other approach. But it's really highlighting, because it is really easy to sort of think, I know I've done it. In this pandemic, I don't want to be going anywhere, so I can just ignore going to the doctor. But if you've got these chronic autoimmune diseases, it's really key that you're still keeping in regular touch with your doctor. I mean, it's obviously you talk about the pharmaceutical company. I mean, it doesn't hurt AbbVie that the fact that they're talking about diseases that have drugs to treat them. So there's a little bit of a
Well, now, John, to your point, it does say it's unbranded, but maybe I was a little bit cynical today in terms of but it clearly is their major, Humira's, which is anti-TNF, and then IL-23 inhibitors for psoriasis, and then their JAK inhibitor for rheumatoid arthritis. So you're right. It is sort of marketed as you said. I think this is a critical, maybe even a potential another podcast topic, is the impact on just medical and health field in general by COVID, right? So as you mentioned, there's been a tremendous dip or kind of rebound a little bit in terms of doctor's visits, but like elective surgeries and things that actually keep hospitals profitable have really dropped dramatically. And that's hurting our healthcare industry quite a bit. Although I don't know how bad I feel for the surgeon who works a couple hours a day and makes a lot of money. But hopefully hopefully our healthcare system's in good shape. I imagine a lot of them are being repurposed today as we see an increase in ER visits and things of that nature. But anyhow, John, it is a little bit of a you're right. It did kind of portray it as, hey, make sure you're staying healthy, but oh, by the way, here's my drugs, right? So anyhow,
I think, but yeah, I mean, we're sort of being facetious here, but ultimately the message is important that, yeah, you still, we have to be obviously cognizant of the pandemic, but if you've got underlying conditions, please stay on top of those so that those are being addressed as well.
yeah, there's some numbers out there, and I don't want to. I forget. I remember reading it about a month ago. A former professor of mine, he's I think he's at Rockefeller or something like that now, and he was posting the impact. Like one could make a case that the lack of going to see your physician has killed more people than COVID has. But again, that's something we'll just shelve for now.
Sure.
But an interesting topic. John, our next one, right, goes right into some big players in the space here, partnering up. Amgen, Takeda, and UCB joined forces in a new COVID-19 drug repurposing study, which, John, I always love seeing these big pharma companies getting together, although Amgen's more biotech. But yeah,
I think this is one of the big stories of how COVID has changed things. I mean, this is all part of the COVID-19 alliance that we talked about several podcasts ago, where it's an alliance between 20 drug makers. In this case, it's Amgen, Takeda, and UCB, looking at 3 anti-inflammatory drugs, see if they can actually address the extreme inflammatory response that we see for some patients. And so there's a couple of approved drugs being Amgen's Otezla, as Takeda's Tac0, and then the UCB drug is an experimental one, so it's not approved yet, called Zaleucoplan. But I think that this to me is the take home from this is it's the coming together of multiple companies to really focus on a single problem. This is actually done as an adaptive trial, so it's starting with these 3 anti-inflammatories, but if any of them are shown to be ineffective, which sort of mimics the recovery trial, which was done in the UK, that led to the approval of dexamethasone and the rejection of hydroxychloroquine. But if one is found to be ineffective, it gets dumped and something else comes in to take its place. Right. And that may be another, it may still be one of these 3 drug makers, or maybe another drug maker again, because like I say, it's actually an alliance of 20. So it's really good to see, and it'll be really interesting to see if this proves anything. Although, I mean, I have to say, we're not really seeing much success from adapted drugs so far. It would be nice if some of them actually, we found that they did have an application with COVID.
Yeah, it's I like the adaptive clinical trials is a little bit gray to me, but it is I like the idea of like, hey, if it's not working, let's try something else. And that, I don't know how much you know about how chemotherapies got to where they were during the 50s and 60s, but basically that's what they did. They dosed you with 3 or 4 of them, and then if it didn't work, we try these 3, all that. So, I mean, I think it's a little bit in this setting, like to your point though, John, the success of some of these is still a little short of where we'd like it to be. And I think really it's not surprising, just given the way the disease, the COVID infection manifests itself kind of differently in everybody. So it's not too surprising, right? We see people, lungs getting attacked, kidneys, liver, all sorts of different things. But, I do think there is, clearly there's some benefit to these drugs. It's just getting the right cocktail and getting the patient at the right sort of cycle or the right moment is the challenge, right?
Yeah, I wasn't being dismissive of it. It's just, to me, like I say, there's obviously a lot of focus. We just haven't seen that home run that really is.
Well, I think this is,
and I think you're right. It's down to that multitude of symptoms that we're still learning about.
But John, we're still it's still kind of early on, right? Oh, so if this is, in terms of like where we are over the last 9 or 10 months with therapeutic treatments of COVID, it's light speed, right? I mean, we're just bombing.
Yeah. It's easy to forget that.
Yeah.
How rapidly everything is moved.
we're the ATM, social media society now, and if it's not done in a couple days, it's questionable in terms of your effort. So no, but it's really it is something that we'll continue to monitor, seeing all of our legend, big pharma getting together and partnering up. Great stuff, John. And that group of 20 is really kind of pushing aside, I think, the dollars and the revenue and doing the right thing. And hey, I think we're going to dive in a little bit more on maybe where we are with vaccines and so forth. But it's really coming a long way, John. And it's just been great to see as a scientist, the collaboration across academics, biopharma, small, big, large, doesn't matter. Everybody's rallying together, and that's wonderful.
Yeah, I mean, I don't know if you've seen this, and I didn't touch on it and maybe thought about having it as a topic, but it does play into this. But there's actually a study kicking off where it's collaboration between a number of African nations, and again, taking this adaptive trial approach. I mean, based on trials that we've seen in Europe, etc., the candidates don't. I'm not sure that the candidates they've chosen don't seem to make sense because of negative readouts elsewhere. But it's still interesting that again it's this pooling of resources, in this case across the continent, to try and address this. So yeah, it's all about that, people cooperating and maybe we can get something out of it.
I think it's just amazing that you're reading about clinical trials in Africa, John. I think that's as you touched on, maybe it's not the best approach, but the fact that there's resources and effort going into it, that's great. Yep. I mean, that's what it's about. We're not, I mean, we sit here in the US and we're not overly concerned with getting a vaccine, right? But the rest of the world needs it, or we're never really going to be in a safe place anymore, right? So everybody needs to be vaccinated worldwide, or we're not really going to, get this sort of mask wearing and fear of each other diminished. Because someday I'd love to be sitting in a crowded stadium cheering for my hometown team. But John, we're going to move on.
Yep.
The next one here is from the CDC. This is our last COVID topic, right?
Yep.
So the CDC is shortening its quarantine options, and this one I think gets a lot of people a little, I don't know, frustrated, because it went from 14 days, 10 days, now it's 10 to 7 days, they're saying. So this is where people get a little more skeptical around, do we really know what the heck we're talking about?
Well, I think, again, we've got to emphasize we're learning about this as it all goes on. And the 14 days, even if you look to that article, the CDC is still saying 14 days is the optimal time. But what they're trying to do is balance compliance. So they're trying to increase compliance because they think a lot of people are dropping out on the 14 days. They don't have numbers on it, but that's just the perception. Still hoping to help public health, and then reduce economic constraints, because if the 7 to 10 days, if you just go 7 days, there is a 5% risk of a still residual risk of 5% with an upper limit of 12.5%. But after 10 days, that's down to 1% with an upper limit of 10%. So it's really that balance between risk and benefit. I know I mentioned it a couple of days ago. When I talked about this article, my son and his fiance are coming over for Christmas, and so we've really been mulling over how to make that happen. We're lucky that we're near a college town, so with students having gone home, my son and his fiance they're coming over here this weekend, but they're going to be quarantining in an apartment in Ithaca, and then we'll be watching this, be it the 7-10 day window. I think it's because they've already been fairly isolated in Boston before they come over. It'll probably be closer to the 7-day window before we then get together and do things with them. So I mean, it's the risk benefit thing. You need to put some sort of control in place, but so what's the right approach? And knowing their movement and their history, we think we know what's the right timing. But that's really what it comes down to.
John, that's great. I don't think people are adhering to what you just said at the level that you and your family are, and the rest of us appreciate it. I certainly appreciate it. I would take a similar approach, but I assure you, many, many people are not really even worried about what you just said in terms of visiting somebody from another state and then actually trying to adhere to some of what the CDC is saying. But that's also probably why your family and you are very safe, right? So you are kind of adhering and understanding that, look, if we can just have some sort of buffer around even 7 days, we're going to mitigate that risk down to 5% or 1% or whatever. I think they said 5%. And then if you go to 10 days, it's the 1%, right? So
Sure. Yeah.
really, John, that's great. And I think you mentioned the economic piece. I think some people would push back on that and not worry, we shouldn't be worried about that, but it's the reality. I think when you look at economic issues on people, that can be even more detrimental than the actual virus itself. So, John, good stuff from the CDC. They continue to, try to refine and make this as palpable as possible for all of us trying to deal with this pandemic. So, John, that kind of closes out COVID for the moment. We got a couple neat non-COVID stories, and the first one is DeepMind AI breaks through decades-old protein folding puzzles. And John, I love a good puzzle, but I really like a good disfolded protein as well.
This again, this is totally outside my sphere of comfort, but really, I loved this article. It was really fascinating. So basically, it's we all know that proteins fold into a certain way, and that drives a mechanism of action, etc. But the question has been, how do proteins fold themselves into proper shape time after time after time? So there's been a competition to sort of try and model this, and people have been relatively unsuccessful until Google's DeepMind division have worked on this, and they came up with one a program, I assume, called AlphaFold, which was relatively successful. But more recently, they've got AlphaFold 2, which when they did the test, they actually trained on 170,000 different structures. And then when actually going at different proteins in terms of predicting the structure, it showed a median score of 92.4%. So it's really close in terms of what the actual structure was compared to what it predicted. So that has huge implications in terms of modeling for disease and therapies, and it really is just amazing. I mean, I think the incredible thing is they talk about how much computing power they need to do it. It said trained on public protein data on about 170,000 different structures. The AI model uses 128 of Google's high-end cloud computing cores, running them constantly for a number of weeks. I mean, obviously, there's a lot of investment in terms of making it work, but then the upside in terms of having this ability to predict how proteins will fold, etc., is huge. And there's one person who was giving feedback on it, said that puts us decades ahead of where we thought we'd be on this. So I mean, a huge step forward for that field.
Yeah, I mean, it dates back to like 1994 is when they first started looking at trying to predict some of what they're talking about with this Google. What was it? The DeepMind program. So, John, I think it's a topic that is very fascinating to both of us in terms of, for those that don't know, misfolded proteins are dangerous things. They can cause a lot of problems, but they're also that happens all the time. So it's not uncommon for something to misfold, and then your body has mechanisms to sort of, what it's called, one way is they ubiquinate it, and it goes through like the garbage disposal inside of your cells, and they get rid of it. But what the power here is being able to predict some of that opens up a very big field scientific wise. And so, John, we can put a pin in it for now, but man, this is a really, as you said, just the volume of data that they're going through is unbelievable. And it's good to see all this AI coming to fruition scientifically, because this is a pretty big breakthrough.
Sure. Yeah, absolutely.
The next topic, John, enhancing cancer-fighting T cells by manipulating the microenvironment. So I love a good microenvironment talk, John. That's another area that was a real hot topic until COVID sort of came into it, but they call it the tumor microenvironment. And I'll, just as a segue into it, John, one of the key things about taking a biologic is It goes systemically, as you know, but the key is to get it into that microenvironment and have its action take place. And people are designing drugs and looking at new ways to target the microenvironment. So tell us a little bit about what's going on at, I believe it's UPenn is working on this.
It's two groups, one at UPenn and one at China Pharmaceutical University, because they're both looking at two different approaches. Basically, they say that T cells have been effective in fighting blood cancers, but they have had limited success when it comes to solid tumours, which is somewhat down to the environment within the tumour, where it's low oxygen and nutrients. It's basically a hostile environment for T cell infiltration and function. So two groups are taking two different approaches. And they're also looking at two different enzymes. So I think UPenn combined the T cells with a drug that inhibits an enzyme called PAC4. PAC4 actually facilitates a process by which the cells that line blood vessels are genetically reprogrammed, causing excessive growth of the vasculature that feeds tumors. So if they knock out PAC4, then they prevent that. The tumor growth, etc.
That's angiogenesis and angiogenesis. One of my heroes, Judah Falk, was on sort of that pathway, right? Yeah, no, great stuff. If you can starve the tumor, you can kill it, and that's what they're targeting here. They're looking at knocking out the supply chain and starving the tumor off, right? That's what this is all about.
In the Chinese group, it took a similar approach, but it's, again, different approach. They basically saying T cell metabolism depends on the amount of cholesterol in the cell membrane, and so what their approach is to put in the article it says put a backpack on the T cell, which contains an anti-cholesterol drug, avascimab. And so by having the drug in there, that gets delivered, and that then it reduces cholesterol to then alter the mechanism.
Yeah, it's great. It's almost like an ADC type of thing there, an antibody drug conjugate or some sort of delivery of a drug, and that's what it's about. So, John, this is really good stuff. I love to see even companies working with, it looks like academic institutions working with some China Pharmaceuticals. Is that what happened here, or are they two separate?
Two separate groups.
Well, completely independent, but kind of targeting the same approach. Oh, good
yes,
stuff.
it's both going at the same idea, but coming from completely different angles, but both showing effectiveness with the way they're doing it. So it's all very, very preliminary. So I mean, it's way upstream of where we normally function, but still in terms of proof of concept, it's showing that there's potential there.
Yeah, and you know, this even you hit something that strikes a chord with me. The tumor microenvironment is basically what I studied in graduate school. So it's that concept of you have an acute immune response that through it goes from acute to chronic, and that's the tumor actually, as you said, creates this tough environment to sustain, and the T cells will actually get reprogrammed to go from acute producing IFN gamma, TNF alpha producing to chronic IL-4, IL-5, things that actually like prevent. So when other cells are recruited, they're actually getting knocked down by cells that are rewired by the tumor microenvironment to keep any other T cells or any other cells out of there. So really cool stuff. And then don't get me started on how the tumor can put misreceptors up to cause it. And it's a great, still to be determined. I think there's a lot of things I just said that are kind of putative and things that people are still working on. But John, we're gonna wrap up our last non-COVID, and this is a little bit of a investment news, right? So it looks like Bayer is really getting into the gene and cell therapy space by creating this platform to support partners.
Yeah, I mean, we've worked with cell and gene therapy companies, but they all tend to be smaller entities. But now this is, large pharma are definitely getting on board with it. I think the article mentioned Kite as part of Gilead, and Spark within Roche. But Bayer have bought a couple of companies, Blue Rock Therapeutics, who are a stem cell specialist, and Asclepius Biopharmaceuticals, who are a gene therapy player. They bought them, but they're actually allowing them to function autonomously. And I remember when we were bought by a bigger concern at my previous job, it was like they left us alone to run the business how we see fit, but then you have the funding to really help drive your work and research. And it's the bottom line's important, but it's not, you're not not so concerned about it when you're part of a bigger business. And certainly for these where they're pushing exploratory therapies, it's really important to have that resource and backing.
That's great. And it's an AAV gene therapy product. And you mentioned Blue Rock, who's really probably one of the longer living gene therapy companies. They've been around at least 10 years, I believe. They've made it quite a long time, but now with backing from Bayer, they are able to use that brand and have some deeper pockets. And I think it's, as long as it's a good relationship for everybody, I think it's a great thing. I'm still curious why Bayer wouldn't want to brand themselves, but I don't sit in on those business decisions, but must have some reason why they want to keep the
I think
company's name. Yeah.
I think it's down somewhat to showing other potential partners that here's how we treat these companies when we bring them in. We're not just swallowing them in and make them Bayer. We're letting them be themselves within Bayer.
That's a good point.
That's an attractive thing if you're an entrepreneur who may, like say, partner in the future or sell on in the future, but to know that it's going to live on consistent with your vision is a good thing.
Yeah, no, I get it now. I think that's a good way to tie it together for not only me, but the audience as well, in terms of why behind some of that. So, John, that's going to wrap up our news and notes. We got a main topic today. We kind of touched on it a little bit. Our topic today is around drug delivery, and this is a topic that we've talked about in previous podcasts. We're finally going to dive in a little bit. And John, I think to kick us off, let's talk a little bit about what we call nanoparticles and liposomes, which we both know a lot about. Oh, go ahead, John. Go ahead.
I think probably makes sense just to talk about why this is a topic of major interest. Obviously we're used with a lot of the drugs over the last few decades are mainly small molecule. They are oral delivery, so you take them as a pill. But as we move more into biologics and just more difficult to administer drugs, then there's much more potential for other companies who have expertise in methods whereby you can deliver these otherwise difficult to deliver drugs. We've talked about this in the past where, things like antibodies, you'll have them administered with an infusion. It may take hours in the hospital, but there's companies out there looking at ways that whereby they can either substantially reduce that administration time through a delivery device or make it possible for you to have it in a much more informal setting. So it's a focus to really help the administration of important drugs that, and make it more convenient for people too. So there's a lot of things driving it, but really driving that, how to get these difficult therapeutics into your body easier to make it more effective.
Yeah, and John, to add to that, it's also about reducing toxicity and increasing the potency of the drug, or at least make it longer lasting. That's frequently what's happening with some of these. So it's like a lot of nanoparticles and liposomes allow for slow release of the drug. So again, that's all about the delivery, less time in the hospital, efficacy, and safety. That's why we're talking about drug delivery. So thanks for jumping in and kind of giving the audience a little bit of why this is so important and why it's such a good passion. So, John, with that, we already talked about like nanoparticles and liposomes, John, right? We'll dive in. That's kind of our first sort of salvo at it. So if you don't know what like a liposome is or even a nanoparticle, what that does is it lipids have a very hydrophobic and hydrophilic, and they make what's called a missile, right, John? They form a circle and you get water exclusion and it makes this nice sort of particle. And within that, they can package in drugs or they can link things to it, right? There's all sorts of things you can do from there. And then also similar principles, nanoparticles can take advantage of things that are not detrimental to your body, but you can link in a drug so that it can kind of do its, because the body can sometimes, really clear the drug or even prevent it from being effective if it's not allowed to get systemic, right, John? So
Sure.
that's my little spiel on nanoparticles and liposomes, but you can share your thoughts.
I mean, yeah, you covered it really nicely, but there was a couple of good examples that we found you'd found that we could really sort of describe what sort of applications of this. I mean, we look at the Evotech one, which is basically they've developed a technology to target lower small intestine colon, but now they're looking at how they can deliver to the upper intestine because that's where drugs absorb better, but it's a more acidic environment and further along the digestive tract. So the Evotech approach is a proprietary coating that covers the active molecule, particularly acid-sensitive molecules. And so what they're doing is this next step on is a dual coating. So their approach is by having different releases on the coatings, it then allows the drug to be released within the upper small intestine within 30 minutes of administration. So it's really playing with what can we cover this drug with to then deal with the conditions within the body. And then the great example that you found was this the light activated liposomes from the University of New South Wales. I think you should probably talk through that one because it's fascinating.
Yeah, John. So you said a lot of things. The one in the stomach, I'll touch on that one first, and then maybe we can jump into the CRISPR because I want to emphasize, things like inflammatory bowel diseases, Crohn's disease, a lot of your intestinal diseases, which I mean, 1 in 5 Americans has this sort of disorder. It's a very high number. It's an affliction that affects a lot of people, right? And so things like anti-TNF alpha work quite nicely, but you got to give that systemically. And so a couple things happen when you do that. One, there's some opportunistic infections that can happen because you're knocking out a key inflammatory cytokine. And then the other is you build resistance. Yeah. And so then the actual therapeutic effect doesn't take place. Well, when you can target it in some sort of liposome nanoparticle that's designed to get into the intestinal area and not get killed, right? Not when I say killed, or not get destroyed by the high pH levels and some of the stuff that goes through the stomach, right? That's what a lot of these technologies are, to get into the site of where the TNFs kind of at a high level and causing all this inflammatory bowel disease. This is a great sort of breakthrough for people that experience this because it's not really curable to date, and a lot of them fall into like a very wide bucket. It's called like inflammatory bowel disease. That means the doctor doesn't really know what's wrong with you, right? Yeah. So that's a great one. And so John, I don't know if you want to comment on anything there, and then we'll jump into I love a good light CRISPR or anything in this space. So, but John,
I don't have much to add. Go with the CRISPR, and we'll take it from there.
So yeah, like these things are really cool when so it gets into high. To me, this is like where it's almost like a Hollywood movie when they talk about these types of things. It's like sci-fi stuff, John, to be able to deliver an RNA or DNA drug or some sort of gene therapy. And then after it's in there, all you got to do is like pop it with a light and it clicks on an enzyme that turns on your DNA and can magically cure you of a disease. This is like really cool stuff, right? So I think this is some fascinating stuff. Of course, I forget what CRISPR stands for. I can look that up for a minute, but it's
I can never remember either.
Yeah, but it basically is allowing for splicing and dicing of your genes, augment your T cells and your immune system to attack a tumor. And that is like, to me, someone who's been doing it for over 25 years now, that's like the holy grail of science, right?
Yeah, I mean, it's like this. We talk about ADCs. This does parallel the antibody drug conjugate approach, doesn't it? But this time it's the CRISPR inside the liposome. It's the liposomes there to get you to the target. And so basically, yeah, it's this light sensitive liposome it gets to the target, it sits there because it's holding your CRISPR payload, and then the light's shone on it to then activate the CRISPR.
It's like a Trojan horse, right?
Yeah,
You said that very nicely. I should have started there. You've got this liposome, again, that's our drug delivery, that now it gets, and it can get to its place, and they make it and design it in such a way that it's highly resistant to any of the enzymes and acids in your body and so forth, or pH changes in your body. And then boom, you hit it with a light and it releases it, right, John? It's great stuff.
So just again to emphasize, because this is a long way off humans. It can work up to a centimeter below the skin, although they think it could work beyond that. They can definitely guarantee a centimeter below the skin, but they so far shown proof of concept in cell lines in zebrafish. So animal studies are next step in this. But it's all, again, it's indicative of how much attention there is to delivery and how it can transform. Getting drugs to their target is so critical. This is, again, we touch on ADCs. It's their ADCs are a hot topic of interest because they are they're delivering a toxic payload to the site of interest to reduce side effects for the patient. That's the problem with so many oncology drugs, that the actual, the drug itself, when it's administered orally, there's an awful lot of side effects because of general toxicity to the body. The ADC, by getting to target, is trying to hit just that tumor so that the healthy tissue isn't affected by it, and your side effects are reduced.
Yeah, John, it's this type of stuff when you see zebrafish, and I'm sure there's some Drosophila, our good friend, the fruit flies, probably being worked on, but you can't get to the next level without proving it here. So this is at an early stage. You're hearing it live on our podcast for the first time. No, I'm just kidding. But no, it's great stuff, John. This is the stuff that leads to where the ADC field 15, 20 years ago is where they are here with this CRISPR nano liposome, it's a photophobic liposome. So it's actually an X-ray that they can use to kind of get it to go, right? So
Sure.
it's wonderful stuff. And you said it's a centimeter down, and I'm sure over time they could probably get it to somewhat of a deeper dive to hit you with activating them. But John, this is the stuff that our children are going to benefit from, I think. So it's great.
So, I mean, we can move on to one that really touches on the ADC approach even more. Was there's a group in Technical University of Munich and the KTH Royal Institute of Technology in Stockholm, where they've developed a synthetic DNA transport system for targeted delivery of cancer drugs. So this is creating a nanocarrier using mucin, glycerol, and synthetic DNA, which wraps up cancer drugs in a secure package that can only be unlocked by certain RNA sequences unique to disease cells. So again, this is another one where they've shown proof of concept in cell cultures. And the reason they've used mucins, which are proteins found in the mucous membranes of mouse, stomach, and intestines, to encase the cancer drug. And the hypothesis is, because they're naturally occurring, the body should have an easier time breaking down leftover carriers, and then that's bound together with synthetic DNA, stabilized with glycerol, that then holds the cancer payload which remains within that cage. I mean, there's an awful lot of talk about, if you read these articles, an awful lot of different approaches to making cages to carry a payload, and this is just one way of doing it.
Yeah.
So it basically remains intact until it encounters that microRNA, which, like you mentioned, was unique to disease cells, and then the drug is delivered.
Yeah, no, and you say cage, it's a lattice, right? It forms like a very tight package that allows for the drug to stay intact and prevent any degradation. Mucins are fascinating, John, right? So I don't know, MUC1, right? Mucin 1, are you familiar with that? That's breast cancer tumor stuff, right? So these are they're well studied. Mucins have been studied for decades, and now they're taking what they know and packaging them up. Again, to me, when you talk more about naturally occurring things and mimicking those and putting them in me, I'm a bigger fan of that, John. So
Sure,
this is a
absolutely.
really cool system. Yeah.
Yes.
Um, So what else, John? We're kind of diving into nanoparticles. Maybe well, for a second, let's pivot onto like more traditional. When people think of drug delivery, frequently they think of like devices and implants. And one of the there's been a lot of advancement here. One could make a case that some of what we're talking about is device and implant type stuff. But the most notable one is the insulin pump. I think we should talk about how I mean, we don't have an article here or anything, but I don't know how much you know about that space. But um, now 22 years ago, a friend of mine that was in residency looked at me and said, if you want to do biotech, you should be researching insulin pumps. This is going to be man, they need these things so bad. And sure enough, John, they've got some great, there's an advancement in devices where they have either patches on someone's arms for low delivery, or even some stuff you've seen on maybe someone's hip or something, or there's like some subcutaneous implants so that people aren't having to, take an injection a couple times a day. There's where part of the field and some of what we're benefiting in what I'll call the immuno-oncology space is stemming from, where the insulin devices and implants really sparked, I think, a lot of this. But
Yeah, I'm glad you touched on that, because I picked up something else from a couple of months ago. I mean, the diabetes is obviously a big concern across the globe, and a number of the approved drugs for diabetes are larger. Well, the more peptidic, insulin's a molecular weight of 5,700, but the larger drugs, so that again they're not oral administration. But there was basically Novo Nordisk, who are a Danish pharmaceutical company, bought Hemisphere Technologies, and I worked years ago on an Hemisphere project, and again the Hemisphere, they have a proprietary technology, which their approach is to it's called the Elogen drug delivery system, but it's to facilitate the oral absorption of molecules without altering their chemical form, biological integrity, or pharmacological properties. And this has been applied in the diabetes space to make these these compounds more orally available. And
yeah,
like say, Novo Nordisk and Hemisphere had worked together prior to this, but now they've formally purchased them, but showing the potential of this, because Novo Nordisk have a huge interest in diabetes
yeah,
as a medical need. And I've got a quick Novo Nordisk story. So I went on a visiting a bunch of customers around Europe. This is several years ago, and got in to visit Novo Nordisk. And I was coming as an LCMS person to talk about some of the insulin work that had been done by the group that I was associated with. And our LCMS study could completely match what could be done with ligand binding, but also had additional specificity that you and selectivity. So in terms of developing an assay, if there was a single amino acid change in the insulin, we could easily work around that with our method. So I did a presentation to this team, but Novo Nordisk had such a completely different approach, where they were so invested in the ligand binding approach. It was very easy for them to come up with specific antibodies, such that they would invest the time and take that approach to then develop specific assays via ligand binding, where for us it was a easy pivot via LCMS, but they were so invested in different direction, they actually weren't interested in talking to me any further.
well, you got a room of people that were probably like me 10 years ago in terms of, yeah. So I think it was important to touch on that. And there are other implants that are having some impact, but John, we're kind of getting a little long here, but I think we touched on, the impact of the medical device and drug delivery field on COVID. We talked about those advancements in some of the nanoparticles and things like that, and then of course we touched on the CRISPR. So any last thoughts on impact on COVID before we go into like
Yeah,
emerging technologies and some of the stuff I like to
yeah,
think
sure.
about?
The one last thing, this is the COVID topic. Is basically Regeneron, who have the antibody cocktail that has had the emergency use authorization. It's a cocktail of two antibodies, casirivimab and indevimab. But this is looking for this a better way of delivering the therapeutic, and they're actually teaming with UPenn to look at how they can deliver this cocktail intranasally. So they're looking at AAV vectors, so looking at the safety and effectiveness for them to introduce the antibodies directly to the nasal epithelial cells and assess protection against disease. So again, we moved away from devices and everything else. It's how can we get and this touches on the nanobodies earlier with the llamas. It's we know that a prime site of infection is the airways. So how can we get the drug into the airways that can possibly address right at the site of action?
So, John, when you were talking about the insulin space, I don't know if everybody realizes it, but your mouth is a hostile environment to proteins, right? Saliva, all of it. So that's why they've made some advancements to allow for oral drugs to kind of overcome some of the pH changes and enzymes in the mouth. Well, the same thing happens in the nasal airways, right? So there's a lot of things in there, most notably mucus and stuff like that. Well, they're designing things to allow for protection of the drug and then subsequent delivery. And of course, we talked about some of the nanoparticles and things of that nature, but also allowing it to get smaller allows for it to evade and get in and do its action better too. So this is some really cool stuff. Again, I love hearing about Regeneron's antibody cocktails and how they're partnering up with people to make them bigger, badder, and better, right? Like that's what this is about. And if Regeneron historically wouldn't be moving at this pace with some of their what I'll call intellectual property, other than we got to get this done, right? We got to figure out how we're going to treat people, right?
We have a need, so we have to address it. Yes,
it's great. I think Regeneron has been a very altruistic company during this whole time, and it's good to see them working with UPenn and getting these. I think the most promising. These are probably some of the most promising therapeutics, and I could envision a day where you're taking something nasal wise to. It makes sense. That's probably where most of our COVID infections come in, is through the nose, right?
Sure.
Because inhaling it, you'd have to get you'd literally have to maybe have someone sneezing in your mouth while it's open. But, you walking down the street or even outside or you're in a room or something, you're going to be inhaling. If somebody, mask or otherwise, you're in a room for a half hour with somebody, you're going to be getting potentially some inhaled COVID particles. And again, if you have something in there, kind of proactively or treat you post, symptoms. Those again are all things to help prevent any sort of real medical issues, right? So whether you're knocking it down, it's all about knocking it down or keeping the level down and keeping it from aggressing, and you won't get sick, right?
Yeah, it's a question: can this be administered as a preventative via this route, such that he's guarding you against possible infection?
Yeah, well, John, this is the one last thing. So emerging technology-wise, we talked a little bit about light. The other one is sound waves. And so this is where, John, I love a good mitochondrial talk. I don't know how much I've said about the podcast, but mitochondria are key players in, I think, fighting diseases, right? They form the ATP of your body. If you can get your mitochondria going and stimulated, it will, to me, it has a non-specific way of dealing with almost any infection. So if you're not feeling good and all the power cells of your body start turning on, that should cause some benefit. And so people are finding ways to, use sound waves. And I don't know, there's something called Bulletproof Radio, which is a podcast I like, and one of the advertisements, you can get your house fitted with sound waves. And I haven't done this yet, John, but it stimulates your mitochondria all day long. And to me, I know that sounds crazy, but it pulsates waves. And there's stories of, it's like, oh, I can see again. My eyes have improved and my diabetes is down. But John, I think there is some truth to sound waves. Because but remember, the body is a lot of Brownian movements and interactions. A lot of that is sound, right? Vibrations. So that there is probably something in there, but I'll talk a little bit about how you can naturally stimulate your mitochondria, but I'll let you touch on or make any thoughts around sound waves.
I don't have anything to add. I think just talk through it, and let's talk about what you found. And
so some of the main stuff we've worked on some of this, so I can't really talk. I don't want to talk too much about it, but some of what I can share naturally. the best way to stimulate one's mitochondria is to take the last 30 seconds or minute of your shower and let the water get cold, and then 30 to 60 seconds of letting yourself bathe in cold water will stimulate your mitochondria. It will probably make you breathe very heavily too. I tend to do when I'm doing it. My wife is like, "What the heck is wrong with you?" Because I'm in there going, because there's cold water pouring over you. But man, you will feel alive, and that's think about it. There's polar dives. I'm not the first one to know that putting oneself into cold water is a good way to naturally stimulate one's body. So that's one way, John, and I'll let you make fun of me if you want.
No. I'm just horrified by the thought of going from hot to cold.
Well, John, it makes shaving real tough. I got to tell you, you do not shave. Don't try to shave after you take the cold water bath for the last 30 seconds to a minute. And the other way is food. There's a lot of foods you can eat that can stimulate your mitochondria. Pizza, unfortunately, is not one of them. So I'm not a big food proponent, but there's some natural oils that can do it, as well as even like caffeine can be a good way to stimulate your mitochondria. And then exercise. Obviously, there's another way to get the body's enzymes going, so to speak. So, John, good. I hope you enjoyed my walk down stimulating my mitochondria. Well, John, this was a very fun episode. We've gone a little bit long, but we got quite a few news and notes, John, right? We got a lot of feedback.
Yep. More vaccine questions,
right?
Shall we move to the vaccine questions, and I'll go first?
Sure.
So Darren asks, what does it mean that AstraZeneca's trial was on a brief stop? What event reason was there for pausing it, and why did it continue shortly after that? There was actually two events on the AstraZeneca trial, and we have to emphasize this is common in late phase trials, that if you have an adverse event, you want to halt to see, is that adverse event down to the treatment, or is it just a natural occurrence? I think I can't remember the condition that one patient showed in the UK, that it was adverse enough, and there was concern that it might be associated with the vector that was the delivery vector for the vaccine, but after sort of assessment of the data, actually the UK and most other places reinitiated very quickly. The US took longer, did a more in-depth assessment, but still in the end gave the green light for the trial to re-proceed. There was one other event that was a patient death in Brazil, but that actually turned out to be on the placebo arm. So that was just nothing to do with the vaccine at all, and that's why it's important to have the placebo arm there, so you're doing that comparison between the effects of being administered nothing compared to being administered the investigative drug, therapy or vaccine, and so it really is. It was a brief stop, but that is just typical of drug trials.
Yeah, John, if it wasn't a COVID trial, no one would even know it had happened.
Exactly, yes.
Adverse events are anything. Someone, you get a needle stuck in you, there's a good chance you get a headache, you get some site swelling. All of those things are could be the potential cause for delaying or stopping a trial because somebody's got some pain and things like that. But remember also, you just touched on a couple things that were important. The placebo group has about the same number of adverse events, right? So there's just sick people in the hospital or sickly people that when you give them any sort of injection, they can have some effects. So there's just a lot of, I hope that John did a good job of elaborating on why it's pretty common practice and stopping and starting. There's nothing to be concerned about, and let's move on from that, John. But I think it's just overblown.
It's also important to say that that there's different levels of adverse events. So I think it's either 3 or 4 levels, and so depending on the extent of them, they'll be designated a certain way. And really, the adverse events are important during a trial because that's going to help drive the labeling for the drug when it comes to administration. So that they when you're watching a commercial commercial on TV for a drug, and it talks about it may provoke this, and this, in terms of various reactions. That's what's coming from these trials. In most cases, it's going to be rare, because if it was anything more than rare, the drug just wouldn't make it to market. But it's pointing out these are potential things that were encountered during the trial that are attributed to the drug, and that's really, it's all part and parcel of drug development.
Yeah, and I don't think I've heard of any true high adverse events in the COVID space for the audience. Not like gene therapy adverse events that can sometimes happen, but again, you're dealing with a much sicker population. So,
Yeah.
John, a good question from Darren. Let's take the next one. Maybe it's Lori asks, researchers warn some COVID-19 vaccines could increase risks of HIV infection. Do you know anything about these tests by these researchers? So there's three articles that Lori provided for us. And, John, we talked a little bit about this one. It's premature to go down this path. This is
Absolutely.
very early. I think to say there's increased risk of HIV is just, the short answer from me is no way. It just can't. And John, I'll share. It's way too early. There's there's coincidences that might have happened, but the idea that you could be either more susceptible to HIV or get HIV from a COVID-19 vaccine is just gobbledygook. Do not listen to any of that.
Yeah, I think it's you're not going to get it directly. That's absolutely the case. I think this came from a letter in the Lancet from a team at the UCSF who said they'd seen some issues when trying to develop an HIV vaccine, that using the AD5 vector, it increased the risk of HIV-1 acquisition amongst men who received that vaccine. But I mean, again, this was in a vulnerable population in vulnerable areas. So if it's a high HIV area, then it might increase the risk of them developing it. But it's not coming from the vaccine. They're raising the alarm about the AD5 vector, but basically no one knows. It's speculative at the moment, and it's one thing to be looked at.
It's just somebody trying to get 10 to 15 seconds of fame. That's what this is. There's no way. I mean, it just is. I can't even begin to fathom how they're trying to link this. If you are at a risk for HIV, I got news for you. You are at a risk for COVID and any other virus, right? This gets into, if you have some primary immune deficiency or some sort of immune deficiencies, you're at risk. And taking a vaccine or not is not increasing your risk. So please, everybody out there that's listening, do not believe the hype around getting more susceptible to HIV because you took a COVID.
Yeah, I think it's basically, again, just as I said, in 2014, NIAID recommended caution when using this vector in vaccines for regions with HIV prevalence. It's only a theoretical. There's no real concrete proof that using that vector on a completely different vaccine is going to lead to this.
If you are unfortunate and you're HIV positive and you're listening to this podcast, or you know someone that's HIV positive, do not tell them not to take this vaccine or any vaccine. That is crazy. I just, whatever collateral damage may be caused by this it's such a low percentage, and I'm disappointed to see it was the New York Post posted some of this. I mean, John, let's move on before I get a little bit upset, right?
The next one is Rodney asks, I know about Pfizer, Moderna, and BioNTech. What other vaccines will be available, and what are the main differences? Also, will we have a choice as to which we should take? So I think in terms of the other vaccines available, I mean, there's more emerging on a monthly basis. If you look at Endpoints News, they have a league table of where the various vaccines are in process. I think they started with 38, and there's they're around that number of investigative vaccines, but there are more emerging. When we talk about the drug delivery piece, I'm still seeing about new vaccines coming through, or people are talking about a different approach. So and you're talking tens of candidates. We've got basically one approved in the UK. I'm sure that the Moderna will be approved globally as well. But as we've talked about, those are mRNA vaccines, which have currently distribution issues because of just having to be kept stored cold. There are other DNA-based ones. There are others that are like inactivated virus-based vaccines. There's a whole host of different approaches. Obviously with investigators around the globe. And we're going to need all these different approaches because for some countries with infrastructure, it'll be easy to deliver these mRNA vaccines with the cold storage demands. But we need ones that are stable at room temperature so that we can administer to developing countries where that infrastructure doesn't exist. I don't know if we'll have a choice about taking a vaccine. It might just be it's available in your area and take it. We do right now. I should have forwarded this to you yesterday, Dom. Someone had done New York Times has done I think it's a link whereby you enter based on 4 simple questions. It's some company who'll be looking at distribution of vaccines, and you enter your age, something else, and whether have you any underlying health conditions, or if you're a key worker, and it says here's where you're likely to be in the list of people getting the vaccine.
Oh, wow.
I think it's only a few simple questions. I was at 287 millionth in the country. And in the state, I was like 62 millionth. If I changed my name to an essential worker, which I'm not, but it's just to do it for my own sort of interest, I think I became the 127th millionth person, and then it reduced where I was in the queue within New York State as well.
Gotcha.
So that, I mean, for most of us who are healthy and low risk, and not key workers, it could be quite a time before it's available. I mean, we're not going to be talking getting the vaccine next month. I mean, we're hoping that by midway through next year, the vaccines will be widely available, and I don't know which ones are going to be available in which area and how that will be distributed.
Yeah, and so I won't add too much. The one thing you touched on, the attenuated virus, that's a protein based, and those are the next wave. Protein and DNA based, to answer Rodney's question, those are coming. GSK's and Merck of the world, a long legacy of vaccine history to them, and they're coming down with the attenuated viral or some sort of protein based. And then in terms of will you have a choice? This is I like you said, not today, not in 2021, probably not. If I'm sure you could always navigate your way to a certain vaccine if you are patient, and I don't know, maybe if you're at least do a lot of research and find the right doctor. But overall, if someone asks you to roll up your sleeve and take a vaccine, if it's Moderna's, Pfizer's, GSK's, just do it. Like you said, John, you and I are in a category where we're going to be like the 300 million person to get it. But the moment that comes, John, I'm there. My arms up. I'll take the Moderna one. I mean, if you want to wait to get a choice of it, then you can wait. But I don't know. I'm ready to feel protected. I got COVID fatigue going on, so I'm ready to take a vaccine and go, try to get on with my life. So a couple more questions here, John. Jennifer asks, what has been done to remove toxins from the fillers in their vaccines? John, that one, so I don't have an answer because I don't think there are any toxins in some of these vaccines or fillers. But if they are, they're at such a low residual level, they don't pop up on any sort of, typically they can't be seen in any sort of test. How they're removed is through just a standard washing cycle. I wouldn't be afraid of any toxins within a vaccine. Whatever's in there is stuff you probably naturally ingest. And again, there's been a lot of misnomer around vaccines. So I hope I answered that one. The follow-up question is, based on your positions per previous podcast, do you have any reservations about the efficacy, and B, public health safety of this rushed vaccine? So, John, I think early on we had, I would say no, I don't have any reservations about the efficacy. We got brilliant people, unbiased people, looking at this data, it is what it is. It's the efficacy. Even Moderna, I think yesterday said they showed 3 months of immunity. Great.
Yeah.
The answer is no. In terms of public health safety of this rushed vaccine, I would caution the word rushed. I don't feel like it's rushed anymore. The amount of manpower, when we were talking about this in the April-May timeframe, we never envisioned the top 20 companies getting together. We never envisioned the collaborative research that's going on. So I don't feel like it's rushed. It's been a focus, and therefore something that takes 10 years. If 10 people are working on something that takes 10 years, if you have 100 people working on something, it can take a year. Right, John?
Yeah.
That's what I'm thinking.
I totally agree that I think the word rushed is completely wrong. It's certainly accelerated because of the way we've applied ourselves. I mean, I think it's you got to realize that when you're hit with a problem, you will find ways to. I mean, what we've seen here are things have been done in parallel rather than sequentially. So we're typically we'd be going through the drug development process and then manufacturing would follow that. We're actually doing manufacturing during the drug development process because the need is so urgent. I'm going to go back to a old example of mine in terms of how you can completely flip your approach on things and accelerate stuff. In my previous position, I actually was approached to run a soil extraction for an experimental fungicide. So I came at this so the typical approach was you obviously extract out of soil, and it's very labor intensive for similar compounds, and the throughput was probably for typical assays around 5 samples a day. I came at this problem knowing nothing about that was the typical approach. I came at it from an LCMS person point of view, thinking I'm a CRO, I need to run as many samples as possible in a day. So I approached it from the point of view of how do I parallel things up? And through various mechanisms and just the way I approached it, we could run 60 samples a day. So it was a real change in throughput. So where we were running thousands of samples, I would complete that work in a much, much shorter time frame than if I'd taken the conventional glass tube approach. I like I say, it was really coming at it from a workflow point of view rather than how do we typically do this. And that's what's been done here. It's really looking at where can we accelerate things, knowing that we have a goal to reach, but we need to reach that goal quicker than we normally do.
Yeah.
And that's the driver here.
And like you said, the whole concept of manufacturing before it was ever really approved, that's expedited things. So
Sure.
I don't feel like it's rushed, John. Maybe we'll move on to Aubrey's question. I think you can go ahead and read it.
Okay. So
It's a big one.
Aubrey says, currently Pfizer's vaccine is being touted as effective for those without prior exposure. What does that mean for those who have had previous exposure? Yeah.
If you want, John, let me help here, right? It means nothing, John. So I think if you have had previous exposure, that's not that I haven't seen anything that dictates that says, oh, if you've had COVID, then you shouldn't get a vaccine.
Yeah.
Now, I don't know where that's coming from. So that one's catching me a little off guard,
and I'm going to add to that. There are limited cases of reinfection, but there are cases of reinfection because there's mutation of this virus. So if there's a vaccine, I would take it. Even if you've had exposure, take the vaccine because you're doing it preventing anything happening downstream. So I would say absolutely you should do it.
Yeah, and then John, go ahead with the follow-up question.
I think I'm going to need you to answer this one as
Sure.
well.
I'll read it then. If the vaccine becomes mandated for things like public school attendance, will an antibody test providing prior exposure preclude the mandate and allow it for an exemption? No. Look, the answer to that is absolutely not, in my opinion. Again, look, this is just Dominic Moreno's opinion. This is not reflective of KCS's opinion. And the reality is there's not a lot of science behind answering your question, other than historically, the answer to that is no. If you still had antibodies against the flu vaccine, the previous year's flu, you still take it. So it's a short answer. Like there is and I don't know if there'll be a mandate, but if there is this like, hey, my child needs to take this to go. He's already tested positive. I don't think he needs the vaccine. No, just take the vaccine.
Yeah.
Because to put it in perspective, it's not a single. Remember, there's many entry points of the virus, and the more protection, the more robust, the bigger the repertoire of your antibodies, the better. How's that, John? Does that make sense what
Yep.
I'm saying?
Absolutely. Yeah.
Go ahead. Ask Mike's last question here.
I think you're still going to be answering this one. Mike asks, has there been any more research into how long antibodies remain in the system? This would seem to determine how often a vaccine might be required. Correct? Oh, correct question mark. Well, I think so. There's two questions there. I'm going to say the frequency of vaccine readministration really comes down to the effectiveness data that we're seeing, and Moderna have just announced that they are seeing effectiveness for 3 months, and obviously they're going to keep continually assess that. So I think that's part of it. I don't know in terms of antibodies remaining in the system. What's been done on that? I haven't noticed that.
It's a combination of things. It's not antibodies. It's your memory T cells
Sure.
and B cells that continue to, as soon as you can establish memory against the COVID virus, then your body has low levels of antibodies circulating. But when you get infected, that's when it kicks in. So it's called primed. And think of like your hepatitis B vaccination. You got 3 of them and you're protected for a lifetime. It's to be determined here with COVID. We think it mutates. We don't know. It doesn't mutate like the flu. So it's still, there's no answer for this right now. But I would think there's no harm in an annual COVID vaccine, John. How's that for an answer? Me personally, I want it annually. I don't care if it's different. And until somebody shows me, hey, there's 10 years of data that says there's established circulating memory to my vaccine. You hear me say 10 years. I just take it, right?
Yeah, agreed. I'm going to be there whatever happens, and even with hepatitis, I mean, there was definitely that. You say it's for life now, but there was a period where it wasn't known how long it was last, so you went for the titer to test to see how you're doing, and then if you didn't show the memory, then you go through another course of it. So I mean, you learn as you go along on this stuff. I think one thing I'd really like to do is just quickly back off on this and say, where do we stand on vaccines? Because there's obviously a lot of concern about it, and we were talking about this yesterday. So I'm of a certain age. I grew up in the UK in the late 60s, early 70s, and I was not a very robust child, but I had chicken pox, measles, mumps, rubella, chicken pox twice. I had shingles, and that's because there was no vaccines when I was of that age. All my kids have had vaccines for chicken pox, measles, mumps, rubella. I'm not sure about shingles, but none of my 3 children have ever had a day off school sick for any of those childhood illnesses. I mean, shingles for me, I was off school for 3 months. I actually was talking to my wife about this because I wanted to confirm that none of my kids had any of the listed what we call childhood illnesses, and she actually talked about one of her university friends who had measles as a child, and is almost legally blind from having had it. Very, very, very poor eyesight as a result. So that's the kind of thing you're wanting to guard against. That's why I'm a total advocate of vaccinations. I talk about the trip I took my son last year. I'm losing track of time, but again, we were given a list of vaccinations that we had to have to be contingent of entering that country, and you just do it. So why are we questioning taking a vaccine for something that could really put us on the road to getting back to what is normal life? So I'll step off my soapbox. I'm done now, and I think we can wrap things up.
Yeah, no, but John, you said such a powerful thing there in terms of we're not that far in age, but because I was a military brat, I still don't like needles because all I did, until I felt like 12 years old, I got stuck every year, needle, but I never got sick, John. All those things you talk about, none of that happened to me. So I feel very at one point I used to think, wow, that sucked. But now it's like, great. I think it really helped establish a lifetime of immunity for a whole bunch of things out there because I was getting stuck up until even carried an ID card until I was like out of college, right? So every year I'd get and if you were seeing a military doctor, man, they were rolling up your sleeve every year and sticking you with something. So God knows how many vaccines I've had, and the reality is they're safe, they're effective. I don't know what some people. Even today, we had someone ask about fillers and stuff. I mean, John, we talked a little bit about that. If you're worried about fillers and stuff, don't eat potato chips and don't eat any organic foods because all sorts of stuff are on there. You just, I think it's all about like your immunity. Talk to your physician. But overall, I mean, these things are safe. And John, you even touched on like, I'm sure if you could go back in time, you'd be like, yeah, I'll take all of these things and miss some of these horrific things that happened to you, right?
Oh, absolutely, yeah. I mean, the cumulative amount of time I was off school was huge, and it was typical of all the kids in my school. I remember, you have the daily register, and like especially during the winter ones, there was always kids off because they've got the flu or the mumps or whatever. We all went through it, so it's very, very different then from now.
John, let's talk about next week. We're going to have some guest appearances, right? Is that the plan for next week?
Yeah, we've got a couple of guests that we're lining up.
Yeah, a couple of our business development directors who are big fans of the podcast, and they'll be joining us. And we'll keep it a little mystery, but it'll be an exciting topic for next week. Just know that we've got a couple KCAS colleagues joining us next week.
Yep,
John, you have a great rest of your day. It's a Friday. We're recording on a Friday this week. So John, have a great weekend, and we'll see you in the funny pages.
Yeah, same to you, Dominic. Yeah, I'm looking forward to 5 o'clock today.
Yeah.